Everyone. Hello everyone, I think this is recording now, so welcome to our little webinar recorded chat type thing. Thank you all for joining us. Sorry we can't actually be meeting you all face to face, we missed that. But here's our little segment, an update from Southampton. So I'm Jay Self, I'm one of the consultant ophthalmologists who with an interest in paediatrics and nystagmus, and I'm also an associate professor at the university, and I run a research team looking at nystagmus, and I work very closely with Helena.
And hi, I'm Helena, and as Jay said, I am also a clinician scientist and associate professor at the University of Southampton and a consultant with an interest in neuro-ophthalmology, pediatrics and nystagmus. And I also want to share hello from the rest of the team at the University of Southampton, which we're going to show you via photo because there are so many of them. So it's a hello from our research orthoptists, our research nurses, the rest of the pediatric surgeons, and the other trainee clinicians, the PhD students, our basic lab scientists, our collaborators, people from other units like the genetics department. There's plenty of people, aren't there, Jay?
There's quite a lot missing from that slide as well, so apologies to all our friends. We collaborate a lot with the other people you hear from today as well, and I think it's only John appearing on our little slide, but there are lots of other people. But the point of this slide really is just to show you that there's a big group of people working on nystagmus, and we're all passionate about it. Okay, so the next thing we wanted to mention was how we've been doing during COVID, because this is obviously the hot topic and why we're doing this by video. I think apart from all the obvious things which is affecting everybody, the two main ways it's affected us is number one, seeing patients, and number two, the research from the seeing patients point of view. Unfortunately, we've had to massively reduce the numbers we get to see in the eye unit, which means that all our patients have been prioritised, they've all been triaged, and so a significant number have had appointments pushed back, pushed out, changed to something else, etc, etc. So first I'd like to apologise to anybody who has been waiting for either a follow-up or a new appointment in Southampton. We have had to triage a lot, so we are essentially seeing just the emergency cases at the moment most of the time. So these are people who will go blind unless we do something in the next couple of weeks or even couple of days. So I'm sure you understand why that's happening. So that's been the main impact on our clinical side. Also, a lot of our clinician friends have been taken elsewhere, so they've been seconded to other parts, COVID wards, etc, etc. But this is the same everywhere. This is not unique to us. The second way, as I said, that COVID's impacted us is on our research, and it's done that in a few ways. Mostly bad, but sometimes good. Our actual lab was shut for a good few months, so a lot of our basic science research, so the cells stuff, the working in a lab with tubes and pipettes and DNA and stuff like that, has really had a massive pause to it. It's meant we've had to focus more on the other bits and bobs we can do remotely, but some of the silver lining is that some of the projects we have are related to COVID in some way, and this is just one example that Helen has put up here. This is a study which one of our research orthoptists, Dan Osborne, is running. This was already sort of preliminarily set up before COVID, but because it's looking at testing kids at home to stop them from having to come to the eye unit quite so often and reducing appointment numbers, getting more data on their vision in the real world, etc. This suddenly became very important because it's obviously very related to COVID in terms of how we cope with the huge demand and how we mitigate for the fact we're not seeing so many patients. So there is a few silver linings, but generally both have been impacted hugely, as have all other parts of life. So the last kind of thing we wanted to talk about really are the projects that we're doing. We're trying to make this not too listy, I'm afraid, but it's quite nice for people to just hear a couple of words on most of the things that we're doing. So I'll kick off, Helena. The first thing we're doing is a long-standing study looking at complex genetics in nystagmus. We know that from our previous work that actually we can do quite good panel testing on the NHS, and we're really proud of the way we've helped push that forward with other people to getting NHS tests much more available and being done finally, which is great. The problem is that quite a lot of those results still come back with partial results, so we're not quite sure if it's caused the disease, etc. So we're working a lot in the background to work out how complicated spelling mistakes or combinations of spelling mistakes cause nystagmus. That then leads into another study that we're doing, trying to improve the diagnostic testing methods used in the clinical lab. And some of these things are about trying to improve the genetics by not having to go back and test mum and dad's DNA samples months after getting one from their child, but actually getting everything we need to know from that child. And without going into too much boring detail, that essentially means that we could speed up the whole process of getting to a diagnosis without dragging it out for 6 months, a year sometimes. The next study Helena's already put up a picture for is looking into improving Electro Diagnostics. So this is ERG and VEP. This is where people have stickers on their heads and lots of wires going to a clever machine and looking at a TV screen, often with stripes moving and funny pictures. A lot of you will have an experience of that, and it's often in a lab like this one on the picture. This is a big set of kit. It requires very specific expertise, but is incredibly useful for diagnosis in nystagmus. The problem with it is that because it has such specific expertise, there's only a few people in the country who can even use it. There's even fewer with the machine, so lots of patients have real problems getting to access these and therefore don't get the test. The other problem with it is that when we do the test, quite often it's not quite right in people whose eyes are moving, so we don't get as much information as we want. So there's 2 studies we're doing on these. Number one, in this picture here, you can see a little handheld device which can be used to essentially get a rough idea of the electrodiagnostics. It's not as good as the full lab, but it's much cheaper, can be done by someone with a few minutes of training, and potentially these devices could be adopted by units up and down the country. So that's one thing we're working on. We just published a paper on that. And the other thing is we're collaborating with Matt Dunn in Cardiff to try and make the full kit better for people whose eyes move. So when you have nystagmus, so we can get more information and improve diagnosis that way. Another totally separate study we have is one looking at mechanisms. So one thing about finding genes for conditions is that it can be useful for diagnosis. But the next question is, well, so what? If you know you've got a spelling mistake in a gene that causes a disease, how's that going to help me? And so the first steps towards developing treatments and working out what's underlying the condition is looking at mechanisms, and we're just publishing a paper on that, and that's on the FRMD7 gene. That's with a previous PhD student of mine called Ahmed Salman. Another study we have is looking at preclinical development of assays to test new drugs. Now, that sounds like a fancy phrase, but essentially it's creating mini versions of patients in a test tube in a lab so that we can test a huge number of potential drugs which might help nystagmus, and particularly albinism for this particular example, without having to test lots of patients with lots of drugs in clinical trials. Because clinical trials, you can only ever test one potential medicine. You can test it on a small number of patients, and it costs millions and millions and takes years and years to do. What's much better is if you can get a little assay in a lab which responds like a little mini patient, but you can test thousands of drugs at the same time and do it very, very rapidly. And on this picture, you can just see a little row of faces at the top. These are patients with different forms of albinism. In fact, the one far left is someone without albinism. That's actually my daughter. You can see she's got dark hair and dark eyes. And when we've made a little cells in a lab that have no spelling mistakes in albino genes, the cells produce nice dark pigment, just like Florence. However, if we produce cells which have a very common variation, which can cause mild forms of albinism, they get a bit lighter. And then as we introduce variations or genetic changes which cause albinism, you can see the cells get lighter and they correspond to the pigment in the patients that the genetic mutations have been copied from. So you can see we've got little mini patients in the lab. Obviously, these cells aren't actually taken from patients. So there's no cells required from patients. These are other cells which we've introduced the spelling mistake to. But this is going to really kick off hopefully just after Christmas when we have a new postdoc starting. So this is a clever scientist who'll be in the lab the whole time looking at this. And we're trying to look at lots and lots of drugs at the same time to try and see which ones have potential to help albinism and possibly other forms of nystagmus as well. I think the last few studies we're going to talk about, I think Helena's going to cover. Is that right, Helena?
Yes, so I'm going to mention the Olivia study, which we've been working on for the last few years. And in this study, we've been trying to replace a chemical called L-DOPA, which is known to be missing from the developing albino eye. We've been able to do this orally, and we have succeeded in proving that we can improve retinal development and visual function in mice with the version of human occutaneous albinism. So I'm going to share a video of that so you can see what happens in a mouse that's been treated with levodopa. Okay, so now that we've shown that oral levodopa can rescue retinal development and visual function in albinism, and we have also shown that this works with human equivalent doses, we are now preparing for our first pilot clinical trial of oral levodopa treatment in infants with albinism, and we're hoping to start this next year, pending resolution of the COVID-19 situation. So all very exciting. And linked in with this, we have set up a oculocutaneous albinism bioresource database where everyone around the country can be recruited to this database, where all of their clinical examination and genetic results are available. And this means that all researchers who are interested in this condition can access this database for research, and we can also quickly identify anyone who might be eligible for this clinical trial, and as well as others in the future. We are also working on the ASOP treatment study, and this is an eye drop that we normally use to treat glaucoma, but it potentially can also treat nystagmus. And linked in with that is the development of a set of core outcome measures for clinical trials, and this is because the traditional outcome measures for ophthalmic visual clinical trials tends to consist of visual acuity, which we all know is not adequate for nystagmus. It just does not capture the full effect of nystagmus on vision. So we'll be looking at things like time to see, eye movement recordings, etc., to see what would make up the ideal set of core outcome measures for future use in clinical trials. So alongside that, we have been working on iPad as a new visual function measure. A lot of work has gone ahead with that, where basically a child can actually do this at home and pick out basically from a set of faces, a particular face, and we can actually assess the visual acuity this way. Jay can update us on where we are on that.
Yep, so it's really testing. It's more than just a visual acuity. It's testing the slow to see phenomenon. So as we know, people in nystagmus, if you give people long enough to read down a visual acuity chart, they can do amazingly well. But if you go out in the real world and need to see things quickly, that's when it becomes obvious there's a problem with vision. So this is trying to pick up on that, because traditionally we've not been able to measure that. So someone with a really good visual acuity of 6/12 or 6/9 or even 6/6 might have significant vision problems, and this is a way of actually measuring it. So it's about how quickly you can see, and also it's face specific. So it's about recognizing faces, which is another typical problem that's reported for people with nystagmus. So it has shown that we can absolutely pull that out, and it can be a really useful measure. We just published that, but now we're working with Maria Theodora in London, and we're looking at longer term data, and so this is ongoing.
Okay, and then the final things that we've been working on is actually educating and spreading knowledge about how best to diagnose and manage nystagmus. So this flow diagram of how to diagnose and assess someone presenting with nystagmus is actually out of a paper prepared by a new group, which we're very proud of, because it was a collaboration between many of the nystagmus experts in this country in order to agree to this flow diagram. This has been published around the world, and hopefully it'll help standardize care so that there's no longer a postcode lottery. That is our hope, isn't that right, Jay?
Yeah, absolutely, and it's also something people can look at themselves. It's not just for clinical doctors. It's if you Google Self and Lee and clinical pathway, or any of the other experts that you'll see that everyone's been involved in it, you'll find it. And actually it's quite useful, because you can have a quick look and see what sort of things you probably should have had tested, or your child probably should have had tested at some point. So it's useful for everybody to look at, because it means you can point people in the right direction. So it's not just for clinicians, it's also for people to help themselves as well.
Okay, and another question we're looking at is why we've never seen an albino patient with age-related macular degeneration. And we're actually going to try and achieve this by looking at the massive biobank data that has been collected. And this is where hundreds and thousands of people have been enrolled into this biobank, and they've all volunteered to have eye examinations and OCT examinations performed. And hopefully this will give us some insight into why having albinism may be protective against the development of age-related macular degeneration. So I think Jay has a final call out for some of his studies this year.
Yeah, absolutely. Well, just the final one, really. We have a new questionnaire study. So we did a good questionnaire last year, and this got published, really looking about at people in the UK who have nystagmus and what their experience of information has been, what their experience of support, visual impairment support, CVI registration, etc. And that really, again, yielded, unsurprisingly, quite a big postcode lottery for everything. So that was quite useful for us to, again, make the point that we need to try and standardise things. We're just doing a new survey now, which will come up hopefully on the NN Albinism Fellowship, etc., and other web pages. We're just finalising at the moment. And the title of it is going to be "How much do you know about this condition?" And the reason is that we want to recruit people who know about nystagmus, so people with it or family members, but also want to recruit a whole swathe of the general population, just to see how much people know or how much they would assume they know or guess when they come faced with somebody with nystagmus. So it's a really useful questionnaire survey, which I think will help us do a lot of things. So if you see it come up, I'd be really grateful if you could A, do it, and B, share it with as many people as you can, ideally as many people as you know who know nothing about nystagmus, if you can find such a person. So that's my final shout out and call, but it will be coming up soon. Hopefully in the next few weeks it will be available.
Okay, so I think we'll be on hand to answer questions later, and I think that is a goodbye from us at Southampton, and hopefully we'll be able to see you all at some point face to face.
I think so. Thank you. Goodbye, everybody. Stay safe and stay sane.
Stay safe, everybody.
Bye.